Hi, today we're going to talk about statistical hypothesis testing for studying genomes. In particular, we're going to look at this particular thing called genome-wide association studies. Remember, in machine learning, data often comes as vectors. So each column is a different feature, and each row is an example. This is a made-up data set on some sort of employee database. And often we want to do something like linear regression. Okay, so on the x-axis here, we have age, and on the y-axis, salary. Okay, and the 5 blue dots you see here are the data points that come from the table in the previous slide. Given this data, we call it training data, we're often interested in the salary at an unobserved age, for example, at 60. Okay, and to do so, we fit a line on this curve, and using that line, we can predict on the unobserved salary. The thing, though, is that very often, if we're trying to do this in genomics, we have this problem that we actually have too many features. Okay, this setting is known in statistics as high dimensional low sample size. Often called the big P, small n. In this particular case, P is the number of features you have, and N is the number of examples that you have. Okay? You can think of this as deep data, fat data, or wide data, whichever. Okay? In the standard settings of machine learning, we have so-called big data settings. We have lots and lots of examples, so the N is very big, and we have some fixed number of features, P. And the matrix that you have when you have that kind of data looks tall, like the matrix on the left. But in genomics, very often what we have is lots and lots of features. So the P is very big compared to the N, and the matrix is very wide. Okay, and somehow this problem is unavoidable. Okay. For example, in many studies that we have, if you want to look at the variation between individuals to study some disease, which is what we'll talk about today, you often have something like 1 million features, and you look at a cohort of like thousands of individuals. The technology for genome sequencing, or for DNA sequencing to be specific, has increasingly become cheaper. And so the amount of features that you have really is growing a lot. Okay. A good example is, for example, in medical studies, there's a study called PCOG, which is a huge study involving many, many different institutions. Each person in that study has generated 285 gigabytes of data, and there's only a few thousand people in that study. Okay? One other reason that this happens a lot is that in medical studies, diseases could be rare. And even if they were not rare, it's actually quite hard to share data between different institutions in different countries. Okay, this issue also arises in all sorts of other different areas, in ecology, in agriculture, and microbiology as well. Okay, so by and large, in the genomic setting, very often we have this high dimensions, so many features, and low sample size, and this is kind of hard to avoid. Okay, but how does this affect us? Okay, let's go back to the real basics. Imagine we have some matrix, which is of this wide kind. And in this particular case, I've drawn a matrix with 2 rows and 4 columns, right? And if I want to do something like linear regression, I need to solve this linear equation with 2 equations and 4 unknowns, okay? And the 4 unknowns here are x1 to x4. And the labels are written on the right. Now, if you recall, this equation here is something called underdetermined, okay? Because we only have 2 equations and we have 4 unknowns. There's actually infinitely many settings of the unknowns that will give us the answer we need, okay? So this is quite tricky, okay? To have more details about this kind of setup, have a look at the textbook, Mathematics of Machine Learning. Okay, so now let's get to the topic. To be able to tell you more about what genome-wide association study is, bear with me a little while I give you a short tour of genomics. Okay. The key thing that people study in genomics, sometimes called the genotype, is something called single nucleotide polymorphisms, or sometimes called single nucleotide variations. Okay, these are affectionately called SNPs, SNPs, and what they are is a variation in the base, a single base at a particular location in the genome. Okay, so for example, I might have a sequence AAGCCTA, and somebody else will have AAGCTTA. Okay, these is a variation. Okay, it could also be a variation in the single person on different cells, for example. Okay, so given that we have these two possible sequences, we say that there are two alleles, okay, C and T, at that particular location or loci. Okay. Now, the variant, the nucleotide, the C, for example, that occurs more commonly in the population, so not in a particular individual, but if you look at the population of individuals, the allele that occurs more commonly, it's called the major allele, and often written as big B, and the one that's less common is called the minor allele, called little b. Okay? If you think a little bit more about it, actually there's more than two possible alleles because there's more than two possible bases, but by and large, we tend to look at major and minor alleles only. Okay, and then there's this other word, which is, as we know, that humans have two chromosomes, not two chromosomes, sorry, two chromosome sets, and we call it diploid organism. If you have one chromosome set, called a haploid organism, and there are organisms like wheat, for example, that are called hexaploid organisms. Okay. Okay. So now we are in the position to define what a genome-wide association study is. You're given some genotype, which is a specific combination of alleles. So you look at different locations on the genome. Each one is either a major or a minor allele, and you just collect them into a matrix. And the phenotype, which is the trait or the characteristic that you are interested in. For example, whether somebody is healthy or sick. Okay, that's the phenotype. Okay. Now, the kind of study that we'll talk about today is something called case control studies. Okay, you go out and recruit a cohort of sick individuals, which we call the cases, and healthy individuals called the controls. Both sets of individuals are genotyped, so you measure all their alleles, and you record whether they are sick or healthy as well, and that's called the phenotype. So the binary phenotype, or the label, if you like, as a machine learner, is also recorded. Okay. Now, recall that we actually have more genotypes than we have individuals in the study. So what we often need to do is to do some sort of feature selection to reduce the number of genotypes. And today we'll talk about how to do statistical hypothesis testing to do that. Now, what I'll describe today is very, very classical statistical hypothesis testing. Okay. So given a case control study, we want to test whether a particular SNP is associated with the phenotype. So say here, you're interested in tall poppies. You want to check whether the particular SNP that you see, the particular genotype that you see here, indicated by the little dashes on there, is contributing, is associated with the tall poppies. Now, we're very careful not to call this causal. Okay, so we're careful not to say that this genotype causes tall poppies or not. It's really a test of correlation. Okay, so what we do is we look at each SNP one by one. You look at each column in the matrix of data and test to see if there's a difference in the alleles in the cases versus the controls. Okay, if this difference is statistically significant, then you say that the SNP is associated with the phenotype. Okay, so the general framework we'll do is hypothesis testing. I'll first talk about the general framework itself, then I'll go through a very specific example. Okay, so we define something called the null hypothesis, which is that the genotype is independent of the phenotype. Now, this is often the source of confusion because somehow the null hypothesis, which is the thing that you really want to define, is often the thing that you're somehow not really interested in. Okay, so remember, we're trying to select genotypes that are somehow interesting and useful, but actually the null hypothesis are the genotypes that are independent of phenotype, which means they have not correlated at all with whether somebody is sick or not. Okay. The alternative hypothesis is that the SNP is associated with the disease state. Okay. The hypothesis test is essentially a test about these parameters where, which I denote by theta here, whether something is, for some range of parameters, you're in the null hypothesis where the genotype is independent, in some other range, you're in the whole alternative hypothesis. Okay. Now, being able to represent the intuitions that we have into this kind of probabilistic model is the main step that you have to do as a scientist. Once you have done that, most of the time you can find some classical approaches to try and answer your question. Okay, and there are many different textbooks to help you do this. Okay, you need to answer questions like which test statistic you should use, what's the distribution of the random variable that you're interested in, and what is the level of significance that you're there. Okay. For defining a hypothesis test, we need to define a random variable x, and this random variable has a certain range, curly x, and usually on the right side of this, we call it another range, r, called the rejection region. Okay. If the object that we measure, x, is in this rejection region, then we reject the null hypothesis. Otherwise, we don't reject the null hypothesis. Okay. So usually rejection regions are, so you can think of this range curly x as the real line. R here, the rejection region, is some value greater than C, the critical value, so on the right side of it. And T here is the key thing, which is the test statistic that you're interested in, that you try and measure in real life. Okay, so the p-value of this hypothesis test is the probability of obtaining a test statistic as extreme as the one that was actually observed, assuming that the null hypothesis is true. Okay? This is critical. So let me say it the other way around. Given that the null hypothesis is true, so given that the SNP is not associated with the disease, right? The SNP is independent of the disease, that's the null hypothesis, then what is the probability that you observe the number that you see? Okay. Now, there are four possible outcomes of this test. So the null hypothesis could be true, or the alternative hypothesis could be true. These are the two rows. And you could accept the null hypothesis or reject the null hypothesis, depending on the test. Okay. Now, if the null hypothesis is true and you accept the null hypothesis, this is correct. Or if the alternative hypothesis is true and you reject the null hypothesis, this is also correct. And then the cross terms are these two errors. And I've never been really able to remember which one is type 1 and type 2. And the way I try to remember this is by trying to recall the boy who cried wolf. Okay, so the story of the boy who cried wolf, the villagers first did a type 1 error, and then the villagers did a type 2 error in that order. Okay, first, they didn't believe. So when the boy cried wolf, but there was really no wolf, and the villagers kind of made a type 1 error, they believed it falsely. And then when there really was a wolf, the villagers didn't believe it later. Okay. Okay. The probability, the level that you choose is called the significance level, and this is often chosen to be 0.05. Okay. This worth belaboring a little while. The p-value is something you compute from your observation, and the significance level you chose a priori. Okay, so let's go to a concrete example. Let's assume we're looking at one particular location in the genome of a haploid genome. So there's really only two possibilities. Okay, and we look at this study with 200 individuals in it, 100 of whom are cases and 100 are controls. We assume for this particular case that the two alleles, the major and the minor allele, big B and little b, they're equally common in the population. Okay. So this means that the null hypothesis, so if we say, so we set the null hypothesis to be the fact that there's no association between the allele and the phenotype. Okay. Now, if that's the case, we get this table on the right here, which is the expected table. Okay, there's no association between the allele and whether there's a disease or not, and so you get the two rows to be equally distributed. And because we also assume that the major and minor alleles are equally common in the population, then the two columns are also equally distributed. So you get 50 in each entry of the table. By the way, this table here is often called the contingency table. So we name these variables so that we can refer to them later. E for the expected value of this, and there are four possibilities, and we just annotate them, whether the cases or controls, cases being 1 and control being 0, and major and minor allele, big B and little b. Okay. Okay. So now we've got the expected table. We go out to the field, we look at our field of poppies, and we see some poppies are tall, some poppies are not, and we genotype them, and we observe the following table. Okay, so there are 23 cases with the major allele, 77 cases with the minor allele, and so on and so forth. We label these numbers here as O for the observed values, and again, indexed by the big B, little b, and cases being 1 and control being 0. Okay. Now, we want to check whether this table this table here is different from the expected table. Okay. To do so, we just measure the difference between them. Okay. And we measure this difference using something called the chi-squared statistic. Okay, and the test is called the Pearson chi-squared test of independence. Okay. How do we measure this? Now, this sum looks really horrible and big, but actually there's only 4 terms in this sum. It's literally the differences between the 4 values in that table. Okay, you take the squared difference, squared because sometimes the observed value can be bigger, sometimes can be smaller than the expected value, and you normalize it by the expected value. Okay, so you compute the normalized distance and you get some number for the observed. Okay, turns out that this variable x squared has something called a chi-squared distribution. A really scary looking distribution, but for all practical purposes, you either look it up in a table or any modern computing system will have a number, a function that will allow you to evaluate this. Okay, so now I just copied those 2 tables here. So you have the expected table on the left and the observed table on the right. And you want to check whether those 2 tables are different. To check whether they're different, you calculate the chi-squared statistic. And I've just written out that calculation here. And you get 42.12. Okay, so is 42.12 big or not big? That is the interesting question. In other words, what is the probability of observing a value that's greater than 42.12 given that the null hypothesis is true? Okay, so remember, if the null hypothesis is true, then you expect the table like on the left, but you've observed a table on the right. And given that the null hypothesis is true, what's the probability of observing a table on the right? Okay, and you measure the distance between the two tables using this normalized squared distance, and you get 42.2. You look up the table of the chi-squared distribution, and you can see that the probability that x squared is greater than 42.2 is actually 10 to the minus 10. Okay, so we can conclude that this statistic is actually very far out in the tail of this chi-square distribution. Okay? So it's very unlikely to see that observation. Okay. The p-value often tries to, people often try to find intuitive descriptions of p-values, and it's worth just writing out what it's not here. The second last bullet point is something I'd like to stress here. The p-value just basically tells you, the probability of observing that value. It doesn't tell you the size of the effect. It doesn't tell you the importance of the effect. It doesn't. It's not indicating that. It's just how surprised you are by measuring that if you didn't have any association. Okay, so that's all for the statistical hypothesis testing. Now, remember, that was only for one locus. Okay, so one particular location, one particular SNP. As I said at the beginning of this talk, very often you have millions of these things, and you want to do this at every column of your matrix. Okay, and you face this issue called multiple hypothesis testing. Okay, so you're doing the test again and again, and like many things, this is well described by XKCD, where somebody thinks, okay, does jelly beans cause acne? You do a test and you go, not really. But then they go, what if we tested every different color separately? So the little picture on the bottom left here tests that for every color, there are 20 colors, and you find that accidentally, actually, that, the green one, you get a positive test. And they then report in the news that green jelly beans are linked to acne. But actually, by repeating the test, each time you repeat it, you have a 0.05 chance of actually hitting something, right? And if you repeat it, you're almost surely going to find something. Okay, and to correct that, there are two very different approaches, which I'll only briefly touch on. This is a very important area of research. One is that you control something called the family-wise error rate. Let's say you have M hypothesis test, so M here says 1 million. Your significance value, alpha, should be reduced by M. Okay, so this is called the Bonferroni method. You only reject the null hypothesis for 1 million repeated tests if the probability you find for each test is less than alpha, 0.05, divided by 1 million, which is M. Okay, and this guarantees you that the probability of rejecting any null hypothesis is less than or equal to alpha. Okay, to see this, essentially the proof basically relies on something called the union bound. Okay. Alternatively, in recent years, people are saying that the family-wise error rate is very strict. It's true, the union bound is somehow the worst case scenario. There are new methods which look at the false discovery proportion. Okay. False discovery is that if the null hypothesis was true, the value V here in the table, but your null hypothesis was rejected, so you think that those SNPs are associated with disease. Okay, so that's the number of items, V, and you divide them by the total number of items, number of SNPs where the hypothesis was rejected. So this is V over R. So this is the false discovery proportion. You can prove that the expected value of the false discovery proportion, which is called the false discovery rate, is then bounded by alpha. And what is this procedure that you do? Well, for a given significance level alpha, remember we have, say, 1,000,000 hypothesis tests, you essentially draw a horizontal line, which is K of slope K, and alpha over M. Okay, and then you sort your p-values in increasing order, and you find the largest value K that crosses this line, and then you reject all the way up to K. Okay, and if you do this procedure, you will find that for a given significance level alpha, the Benjamini-Hoffman method bounds the false discovery rate. That was all I wanted to present today, the basics of hypothesis testing applied to genome-wide association studies. Okay, I very, very briefly introduced things like genome-wide association studies, single nucleotide polymorphisms, what was an allele, humans have a diploid genome, and you collect data. The matrix is a matrix of genotypes, and you have labels called phenotypes. Okay, we introduced this idea of hypothesis testing, where the null hypothesis is somehow the counterintuitive notion of that SNPs are independent of the phenotype, versus the alternative hypothesis, which is that the SNPs are associated with the phenotype. Okay, then we designed a test statistic, and we looked at the example of the chi-squared statistic to check whether the two contingency tables, the contingency table of the expected values and the observed values, whether they're the same. And we can compute the p-value, which is the probability that we observe this test statistic given that the null hypothesis is true. Okay, and you reject the null hypothesis if that number that you compute, that p-value, is less than the significance level, which is called alpha. Okay, if you do repeated tests, like you would usually do in genome-wide association studies, you do it for each column, you want to do some sort of multiple testing correction. Thank you for your attention.